Welcome to the meeting, and can everyone hear me okay? Yes. So delighted to have everyone here. We have over 300 people joining the meeting at this stage, and considering what a challenging time this is for us, we're very pleased that people are taking a little time to think about science while everything else is going on around them. My name is Hugh Davis. I'm director of the NSF Center in CH Functionalization, and we're delighted to be able to host this meeting. Next slide, please. So the center involves 22 professors from 15 universities that have been working together now for several years. And one of the things we had to learn early on was how to handle really large-scale communication. I think this is something that all of us are going to have to do over the next few months to deal with the current situation. And having learned that it is indeed possible to communicate very effectively in a large scale like this, we decided to begin having virtual symposia to really communicate the exciting research that's going on in this field, some from the center and also from the great contributions that's happening all over the world. So next slide, please. So when we started this meeting, the idea was that it was supposed to bring a community together. And we actually encouraged people, instead of joining individually, to actually join in groups send us these photographs of them coming together from all over the world to listen to this meeting. So this time it's a little different. We still want to bring people together, but we're bringing you all together, hopefully individually, rather than as individuals, rather than coming together in a room together. So we can still connect to each other, but now in a fully individual virtual engagement. So next slide, please. And I'm delighted for this particular meeting that we've got three amazing speakers. There's going to be a really exciting presentation for all of us. Now, one of the disadvantages of not being in my university for this meeting is I couldn't go down the hall and check how does David pronounce his last name. So I had to go. So we got David Nichevich, close maybe, Nichevich. We're well, not very close, I don't think. Even though his name is hard to pronounce, he's extremely well known. He's from the University of North Carolina, Chapel Hill, and we're going to hear some exciting work on photoredox catalysis. We're also going to hear from Hans Renata from the Scripps Research Institute, who's got a great combination of expertise in biocatalysis as well as large-scale synthesis strategy. And you're going to see how that's going to come together very nicely in his own program. And then finally, we'll have a talk on using organocatalytic approaches for doing atom transfer CH functionalization, coming from Mike Linsky at the University of Virginia. So three really exciting talks, and we're very delighted that they've been willing to speak to us at this time. Thank you for being so willing to still accommodate this symposium. So next slide. Before we get going, there's a little bit of housekeeping I need to mention. So first of all, during the talks, only the speakers will be able to speak, because obviously we've now got 400 people attending the meeting. So obviously there'll be a lot of background noise unless there's a lot of muting going on through the talks. But if you do want to ask questions, then the way to do that is to log into the Zoom and go to the question and answers at the bottom of the page. And then you'll be able to enter your questions to us. Alternatively, if you're on YouTube, you can go to the tweet question and tag NSF CCHF. Now, one of the things we used to really encourage for these meetings is that when you're meeting as a group, to take a picture. We used to make these collages of photographs of everyone that's been involved in this meeting. So this time we still want a picture, we still want a photograph, but we want a different one because a lot of you are individually and a lot of you are at home. So the photos we want this time is of you and your pet. Okay, so if you've got a lot of chemists seem to have pets, at least in my research group. So we'd like to have the photos of you and your pets. So please send those and forward them to this to the NSF CCHF. And then finally, it's very valuable for us to get feedback from you. So you can do that using the indicated link, but also much more easily would be just to scan with your camera the QR code, and that would take you directly to a survey that you can respond back to us of what you think of this particular meeting. And finally, I encourage you to follow this future meetings that'll be coming up on Twitter, Facebook, Instagram, and LinkedIn. And hopefully you'll be able to join us for the future virtual programs we'll be having. Okay, so if we go to the next slide, just want to bring you up to 2 upcoming virtual symposia. So our center does collaborate with the Division of Organic Chemistry and supports their virtual symposia. And they have one scheduled for April 29th. They will have 2 speakers, Margaret Fall, and the second has been selected, but hasn't been fully finalized, hasn't been fully confirmed. So that will be April 29th. And then the next, the next CCHF virtual symposium will be June 30th. And we have 3 quite interesting talks indeed. We have one that will talk about use of big data to analyze site-selective CH functionalization. That'll be Pedro Perez from Spain. And then we have Ang Li from Shanghai Institute of Organic Chemistry, who's definitely one of the leaders in thinking about total synthesis and applying CH functionalization as a major strategy. And then obviously, photoredox catalysis is really important these days. And beyond today's talk, we'll have Robert Knowles from Princeton University describing the impact of photoredox chemistry as well on CH functionalization. Okay, so without further ado, let me introduce our first speaker, David Nichevich. So David had his BS from UNC Charlotte, then went to do his PhD at UNC Chapel Hill with Jeff Johnson. Then went to postdoc with Dave McMillan at Princeton between 2007 and 2009. And I think it's fair to say, really, he was one of the real developers in that group of the idea of photoredox catalysis. So he was a major player in the initial development. And obviously, UNC Chapel Hill liked him when he was a graduate student because he came back to start his academic career in 2009. Quickly was promoted through the ranks to full professor in 2018. And he's made tremendous contributions to the field of photoredox catalysis, most particularly bringing in organic photoredox systems that had a much wider electrode potential to really expand the possibilities of the chemistry and really highlighted some wonderful examples of applying it to CH functionalization. So David, we're delighted to have you here. And I want to say you are the best dressed speaker we have had in all 17 of these symposia so far.
Thanks, Hugh. That was a really kind introduction. These are my work from home clothes, actually. So, you know, when I come into work, I dress down a little bit. It's a real pleasure to be a part of this, and it's a really fantastic center, and I really appreciate the invitation to be a part of this today. It's almost as if Hugh and the center have had some sort of crystal ball and knew that we were going to be doing presentations like this for a while. It's unfortunate, but obviously we'll press on and do the best we can. So I'm excited to tell you today a little bit about the research in my laboratory. As Hugh mentioned, we're involved in the area of organic photoredox catalysis, and I focused on 2 main projects today that are in my laboratory that involve CH functionalization, two main programs, one in sp3 hybridized functionalization and one in arene CH functionalization. So I'll talk first about our program in arene or in sp3 hybridized CH functionalization by talking about this alpha amine CH functionalization program that we have started. So why are we interested in this in the first place? Well, we saw that forming carbon-carbon bonds adjacent to nitrogens on nitrogen-containing heterocycles and acyclic compounds is a valuable disconnection for accessing, for example, bioactive compounds as well as natural products. Just a few examples listed here. And the way we envisioned sort of going about that carbon-carbon bond forming step is through radical chemistry. And obviously, photoredox catalysis is a great way to generate neutral carbon radical species. And so the way that we envision doing that is by starting with some sort of a protected amine scaffold and then generating a carbon-centered radical adjacent to that amine such that it could react with, for example, an alkene to form that new carbon-carbon bond. Practically, how we hope to carry this out is by using a carbamate protected amine. And so how we are listed below oxidation to now access this alpha amino radical species. Now, again, like I mentioned previously, this really was a mechanistic strategy that had been taken advantage of in the past. However, typically this utilized an aryl amines, and doing this with carbamate protected amines is quite a bit more challenging because you need quite a high oxidizing capacity for your catalyst at about 1.9 volts. And I'll talk a little bit about redox potentials in a second in order to do this one electron oxidation. So that's where this area of organic photoredox catalysis comes into play. And so I often get the question, well, why are you interested in using organic photoredox catalysts? And quite simply, the organic catalysts are much more oxidizing than their organometallic and inorganic counterparts. And so those of you that may be familiar with photoredox catalysis, but may be more familiar with the metal bipyridyl complexes, such as those depicted on the left, these complexes are excellent photoredox catalysts and have fantastically long excited state lifetimes, but they're simply not as oxidizing as these organic counterparts. And to just reference you in terms of some common organic molecules, ultimately, the excited state reduction potential of the catalyst that you use has to be more positive than whatever you're trying to oxidize. So if we orient ourselves in terms of the carbamate protected amine in terms of its redox potential, it's about 1.9 volts. And as we can see, the excited state reduction potentials of these metal bipyridyl complexes are not sufficient enough to be oxidizing, but these organic catalysts are. Why visible light photoredox catalysis? Well, quite simply, we don't want to excite the substrate. Most organic substrates, fortunately, only absorb in the deep UV, whereas these organic catalysts absorb in the visible. And it's advantageous because we can now put all of our light energy into the catalyst and not the substrate. So we've been working with these catalysts for a while. The first was reported by the Fukuzumi lab. This is the mezityl catalyst shown here. These absorb blue photons and readily pick up and give up electrons. So they make excellent one electron
Hey, Dave, I think we're having issues with your audio. I had turned off the video feed to see if that would help, but just so you know, we can't hear you right now.
Yeah, I'm having issues with audio right now. Sorry. Bear with me. Is it connected now?
Yes, we can hear you now.
Okay, I'm sorry about that. Yeah, it seems to be a hardware issue. Hang on. Wait, let's go back here. Sorry. It may go in and out, so just bear with me. I've seen this come up a few times, so I'll try and stop. It usually lasts only a few seconds, so I apologize for that. So we've been working with these acridinium salts for quite some time, and we've also developed multiple derivatives of these in conjunction with sigma. Actually, it should be sigma millipore now. And we find that, for example, you'll see quite a bit from this ditert-butyl-mazidyl catalyst today. That is the most utilized. It's much more robust than the parent Fukuzumi catalyst. And these are some of the catalog numbers for the catalysts that are available through Sigma. Unfortunately, we don't make any royalties off of these, but they're there for use if you're interested. In addition, the Spar lab at the University of Basel, as well as my lab, have reported some much more streamlined syntheses of these catalysts in case you're interested. All right, and so back to the task at hand. With this alpha amine CH functionalization, what we observed is that we could now take just the starting carbamate protected amine, our Michael acceptor, and the catalyst in DCM, and just irradiate these with blue LEDs, and we see very good levels of carbon-carbon bond formation in a number of scaffolds, including cyclic, as well as ones that you might see in a lot of pharmaceutical motifs, as well as acyclic scaffolds, albeit on a little bit lower yields. You can really decorate the nitrogen with a variety of different functional groups, as well as having different protecting groups such as CBZ and FMOC. What's really interesting from the standpoint of using this in complex molecule synthesis is that you can use substituted pyridines and get good levels of diastereocontrol control in these systems. Additionally, you can see that if we start with a stereogenic center, alpha to the amine on the opposite side of the molecule, we functionalize the opposite side and maintain stereochemical fidelity. Now, in terms of the diastereocontrol control, we Sorry, I think I'm back again. We wrap first platinum, where this half chair, the transition state or ultimately this intermediate, the minimized form is where this is a half chair, and this ultimately positions the methyl group in the pseudo-equatorial position, such that through a chair-like transition state attack of the electrophile or the Michael acceptor occurs to give the diastereomer shown on the left. For the compound on the right, if you recall, having a substitution alpha to a carbamate forces that methyl group in a pseudo-axial position to alleviate A1-3 interactions, and again, approach from the top face via first Plattner type transition state gives the diastereomer that's observed. This is complementary, as some of you may know, to a lot of really beautiful lithiation chemistry from the Beat group. And in fact, this exact substrate has been prepared, albeit they observe the opposite sense of diastereocontrol control. So it's a nice complementary approach there. And then just to give you an idea, we've started to apply this to some natural product syntheses. So we can take this actual first transformation here after deep protection of the Bach group with TFA and then a reductive amination. This now gives a single diastereomer of monomorine 1 enantiopure and is the shortest synthesis of this molecule to date. And that's a simple molecule, this indolizidine alkaloid family, but I think it gives you an idea of where you might be able to deploy this in more complex settings. And that's certainly a topic of focus for my laboratory in the future. With respect to the Michael acceptors, really you can get away with quite a bit here. I would say the main limitation here is as beta-beta disubstituted Michael acceptors don't work particularly well in this. And you can scale this up using a flow setup. This is an actual flow cell, 8 milliliter internal volume. And we simply use a peristaltic pump to pump our reaction solution through that. And we've run photoredox reactions like this on decagram scale, multi-decagram scale without any issues. To give you an idea in terms of how we study mechanism in my laboratory, I'll give you a little insight here in terms of this particular reaction. And so if we look at we can look at the single electron we can look at so if we look at the lifetime of the fluorescence, in the absence of any Bach amine, we see the lifetime here in red. As we add increasing concentrations of the Bach amine, we see that that fluorescence lifetime decreases, which is indicative of an electron transfer event. The next step, as I mentioned, is a deprotonation step. And so as I mentioned at the top, ultimately those CH bonds alpha to the amine become acidified, and we were curious as to how much. And so we turned to DFT calculations. And as you see, the starting amine has a pKa of about 30 for those alpha CH protons. And once you remove an electron, removing an electron increases that the pKa of those CH protons by 25 orders of magnitude, which is pretty incredible effect, certainly is known in the literature, but I think it's pretty dramatic here. And what we think is that that now makes those alpha CH protons acidic enough to deprotonate by a Bach amine. Radical conjugate addition then ensues. And then what we think is the key catalytic turnover step here is that that neutral acridine radical does a 1 electron reduction of that alpha carbonyl radical. This is actually a stable persistent radical. We've studied this by EPR. And so we know concentrations of that buildup in the reaction is often the resting state of the catalyst in these transformations. That ultimately would generate an enolate if it does the reduction. And what we propose is that a proton would be picked up from another molecule of substrate. And so to probe this, we did some deuterium labeling experiments. If we take an alpha deuteroamine, we see that with this Michael acceptor, that deuterium atom is transferred to the enolizable position of the melanonitrile. Just to show you that the solvent doesn't play a role in terms of the final proton or hydrogen atom transfer. If we run this reaction in DCM, that's D2 DCM, we see no incorporation of deuterium in the product. So that gives you an idea in terms of the mechanism and gives you sort of a general overview in terms of one of our approaches to sp3CH functionalization. I want to finish the second half by talking about our program on arene CH functionalization. And certainly this is an audience that I don't have to give much of an introduction by, in terms of arene CH functionalization. So we're back again. All right, we're back again. I'll give a truncated in that typical base CH sort of directing group that can ultimately ligate a metal to direct metallation to the ortho position of that monosubstituted arene. And then this organometallic species is then intercepted by the electrophile, which is in solution, to ultimately afford an orthoselective process. Now, where we hope to ultimately complement this is by accessing reactive arene cation radical species by 1 electron oxidation of the corresponding arenes. And as you can see from this charge, this DFT calculation of the charge density map on that arene cation radical, the highest degree of positive charge density here is in blue at the para position to that methoxy group. And so it seemed that we might be able to intercept this with nucleophiles, and certainly there were some indications of this in the literature, again, before we even came along, that suggested this might be possible. And so if we have a terminal oxidant in the pot that can do the second oxidation step after loss of proton, this would lead to a para-selective CH functionalization. And so ultimately, this would be complementary to transition metal based approaches that are generally orthoselective. And then I think importantly, it gets us to new chemical space because we have now nucleophiles instead of electrophiles involved in this transformation. And so I'm not going to spend too much time on this. We published this several years ago now, but ultimately we chose to investigate the use of azoles such as pyrazole, as these are very common in pharmaceuticals. Now using molecular oxygen as the terminal oxidant, we can now form carbon-nitrogen bonds. Again, para to the most electron releasing group. Interestingly, we retain the halides in these molecules, which might be quite a challenge for transition metal based processes due to competing oxidative addition. Heterocycles also work in this context. And then I think from a pharmaceutical standpoint or agrochemical development standpoint, a variety of different azoles work really nicely in this chemistry. And we can do this with much more complex structures that have a lot of different CH bonds that might be susceptible to CH functionalization, and we get good, clean functionalization. And then sterics really aren't as much of a challenge for this reaction as well, which I think is really impressive. Bonds quite nicely. This is the first catalyzation. We can do cyanation, cyanide as our source of cyanide. And then we can use things like primary amines in this case to do CN bond forming reactions. In this case, an amino acid also is reactive in this context. Most recently, we've been able to publish that, for example, diazoalkanes can also accomplish this, which complements a lot of transition metal catalyzed variants in this area. And we've been able to show this on a variety of different arenes and heteroarenes. We've been able to show that other diazoalkanes work in this scope, and that we can do this on pharmaceutical derivatives, albeit the yields are a little bit lower in this case. The last thing I want to show you is that we were ultimately interested in forming carbon-fluorine bonds. And from the standpoint of ultimately introducing fluorine 18 for applications to positron emission tomography. And without going too much in detail into this area, there's been a lot of really nice work from both Tobias Ritter and Veronique Governor's lab in this area. And the prevailing strategy has been to ultimately use, I would say, an organometallic one where ultimately a heteroarene or an arene that's either a halogen or a triflate can undergo an oxidative addition process to generate some sort of aryl organometallic species that is then treated with NF18 source. And just to sort of highlight the challenges here, fluorine 18 is an unstable radioisotope of fluorine that has a half-life of 110 minutes, and it decays by positron emission. Ultimately, we'd like methods that utilize fluoride anion, because ultimately this is how the cyclotron generates F18, and typically this is in the form of potassium fluoride F18. I would say that the pre-existing methods in this area offer some opportunities for improvement in terms of avoiding handling unstable organometallic intermediates. And then also an important thing is if this is utilized in a hospital setting, trace removal of transition metals can be problematic. And then a little bit more versatility with respect to synthesis would be ideal. And so for these reasons, we thought we might be able to target a direct CHF18 bond-forming reaction that is catalytic in organometallic. And so in a really nice collaboration with Ariane, such that we BAF as our F18 source. And at least in this first iteration, we had to go to a fiber optic laser setup that allows us now kinetics fast enough for this to be useful. Although we're in development of a flow LED device, my collaborator and I have started a company, LED Radio Fluidics, that is actually in the process of developing a plug and play device there. But ultimately, we're now able to adapt these conditions, and these are isolated radiochemical yields of pure compound. You might not think 7% radiochemical yield is much to write home about, but for imaging purposes, as I'll show you in a second, that's sufficient. We've been able to introduce F18 label to a variety of different pharmaceutical compounds, as well as the important signaling molecule F18 DOPA. The last chemistry slide I have here is that I'll show you, we've been able to develop some novel probe molecules for a variety of different purposes. This first one uses the non-steroidal anti-inflammatory drug phenoprofen. If we introduce an F18 label here, we asked the question, is this a probe that we can use to look at inflammation? You might wonder why this is important. Well, inflammation of the brain is implicated in a lot of diseases such as Alzheimer's and dementia. So if we now look at this control experiment, this is a mouse here, which we've inoculated with the F18 labeled phenoprofen. This is the CT scan. We see nothing different in terms of the mouse here. We see very little by MRI. But if we now induce inflammation in the ear, we see this now with a significant MRI response, indicating this could be a possible way to detect inflammation. And then the last thing is, this can be utilized as a method for synthesizing new probes for oncology. And so this is a particular amino acid derivative that we label with an F18 fluorine isotope. And cancerous cells uptake resources faster than healthy cells. And so they uptake carbohydrates and amino acids faster than healthy cells. And so we can use this as a probe to look at different forms of cancer. And as you see here in the orange and purple are two cancer cell lines that are indicated in the mouse that we can now develop probes for. So we have a really powerful method for taking aromatic aromatic Okay, so with that, I'll in terms of organic functionalization. I think you saw all the names involved, a lot of whom are here, former alumni that did some really fantastic work in this area, as well as current students in my lab, Josh McManus and Nick Anuska. I'd like to thank these sources of funding, and again, Hugh, for the invitation today. And I'm happy to answer any questions either now or online or via email.
Okay, great. Normally we get the whole room that I'm in to clap, but here it is only me.
Very good.
So we've actually got a big collection of questions. I'm going to ask a couple of them, but I'd like you, if you can, then go to the question and answers to answer some of them offline. But let me ask you a few of the questions. So this is from Nick Chiappini from Princeton University, who's asking, do you see any improved performance of harder to reduce Michael acceptors on the back end with added hydrogen atom transfer donors?
That's a good question. We don't really see a significant improvement with an added hydrogen atom transfer donor in most cases. I can't say that we've tried harder to reduce ones, because that would make the polarity maybe a little bit more mismatched with that. But in terms of the radical addition to that alkene. But that's a good question. I don't know, we've looked at it extensively, but at least with some of the examples we've tried, that didn't seem to help.
Okay, thank you. And this is a question from Nigien Doan, who first of all thanks you for the talk. And given the mechanism you described, that after the radical Michael addition, the radical will abstract the hydrogen from the starting material, does it mean that the reaction is self-catalyzed after the first catalytic cycle? Can we just turn off the light after radiating a while and the reaction still proceeds?
Right, yeah, so good question. Is this essentially a radical chain process? We don't have any evidence that this is a radical chain process. If it is, I would say that the chains are very short, and ultimately you do need constant irradiation. We're not entirely 100% sure of what is doing the final deprotonation step, and if it's a radical based deprotonation or even anionic. So that's something.
Okay, we missed you a little bit at the end of that. David, we didn't quite hear the end of your answer.
Sorry, yeah, it's. Sorry.
Okay, well, Dave, it seems that you've drawn a tremendous interest. You know, normally after you finish a talk, you're supposed to relax, but it's not going to be the case because you've got at least 9 questions online to answer. So we're going to keep you busy for quite some time. So once again, thank you for giving a really exciting presentation. Thank you.
All right, thanks you.
And I want to say we have about 450 connecting with Zoom and about 200 connecting with YouTube. So we're really delighted that so many people have made an effort to join this particular session today. So thanks very much. Okay, so our next speaker is Hans Renata. And in the same way that Dave had made a big impact on his mentor's research, during his postdoctoral studies, Hans has done the same thing, because Hans has got a very interesting background. He had his BA from Columbia University, and then did a PhD with Phil Barron, so really very serious major scale synthesis PhD in 2013. And then he went to postdoc with Francis Arnold. And basically, he played a very pivotal role in the work of the Arnold Group to moving P450 enzymes to do catalysis that are not natural to nature. And basically, I was very excited in what he achieved there, because he was involved in developing novel catalysts that could do carbene transformations. And so with this tremendously strong background in both large-scale synthesis and obviously the major develops in enzyme evolution for novel reactivity, when he started his independent career, he had 2 very powerful skill sets. So in 2016, he's been at Scripps Research Institute in Florida, and has developed some wonderful examples of how you can use enzymatic strategies to really impact significant synthesis of important natural products. And so we're very excited to hear his talk today, which is biocatalytic CH oxidation as an enabling tool for complex terpene synthesis. Welcome, Hans.
Okay. Thank you, Hugh, for the kind introduction and also for the invitation to speak here. And of course, thank you for pressing on with the event despite the difficult situation. Today, I'll be sharing with you recent results from the lab in the area of chemoenzymatic synthesis of highly oxidized terpene nature products. Okay, broadly speaking, my lab is interested in developing useful enzymatic tools for the synthesis of complex molecules. And in this regard, we're especially interested in the development of enzymatic CH oxidation. We realized that there's still a lot of challenges in doing this oxidation chemistry using small molecule reagents or catalysts, because it's really hard to get useful selectivity sometimes, and enzymes, because of their exquisite selectivity profile, we thought that they can be advantageous if applied in this scenario. And we realized that by doing so, we can come up with access to unique building blocks. And if we combine this with emerging methods in organic synthesis, we might be able to come up with efficient synthesis of bioactive nature products. And in the bigger picture, I guess, we would like to show that this approach can be complementary to traditional ways of making molecules, as well as chemocatalytic CH functionalization methods to make complex molecules. And not only that, we also hope to show that we can come up with novel disconnections that ultimately can also be efficient. Now, Phil Barron likes to think that things are better if you include some Elon Musk reference. So I'm gonna try to insert some Elon Musk reference here, and hopefully you will be able to see the parallel. This quote is taken from a documentary that I just watched on Amazon Prime this past weekend. Thank you for work from home. So I quote, "A lot of people have this perception that if you buy electric, it's because you have some sort of electric fetish. And one of the things that is incredibly important is to break that perception and show that electric car can be better than any gasoline car." And I think, similarly, I think that today there's still this perception that if you do biocatalysis, it's because you have some sort of biocatalytic fetish. And I think that it's important that we break that perception and show that biocatalytic synthesis or chemoenzymatic synthesis can offer a lot of advantages over traditional synthesis. And just some housekeeping before we go into the real science. This slide shows a variety of metal enzymes that nature uses for oxidation of small molecules. We work in this space, so we work with P450s and non-heme deoxygenases, or alpha KGs. They have several notable differences. The P450s are heme-containing enzymes, and they also require a dedicated reductase protein partner. This will be important later on in our discussion. The alpha KGs are non-heme enzymes that contain this 2-histidine and 1-carboxylate ion binding motif, and also uses this ketoglutaric acid as co-substrate in the reaction. Now, in contrast to the P450s, they don't require dedicated reductase, and you can reduce them to the ferrous form by using simple reductants such as sodium ascorbate.
Okay, quick thing, there's a little pop-up that's blocking the corner of your screen.
How do I remove that? Minimize like this?
Yeah, build order.
Okay, are we good?
Still there.
I don't know how to remove it. Good enough? I think good enough, no?
Are you able to move it off to the side?
I tried to move it all the way to the side. Is this good now? Better?
It's still showing up the same on my end.
Okay. I don't know what to do.
It says a build order.
Oh. Let me see if I can.
It might be underneath your program.
Maybe let me stop sharing first and try to remove that. 1 second. Wait, hold on. Okay.
Looks
How about
great.
now? Yeah, good. Okay, great. Thank you. Okay, so we became interested in this highly oxidized terpenes. And when we look at these structures, we noted that there is a certain motif that keeps occurring again and again. This manifests in the form of this hydroxy trimethyldecaline motif that's present in either the trihydroxydrimane or the 3-hydroxy N-isocopylane unit in law of terpenes. And you can find this in more than hundreds of terpenoids. Typically, when people try to build terpenoids like this, they will either rely on biomimetic polyinsacchalization or doing a more modular synthesis starting from Wheeler-Mischer ketone. And we thought that we can come up with a complementary strategy to build such terpenes by using chemoenzymatic approach. And our idea here is to introduce this hydroxyl group through biocatalytic oxidation, starting from terpenes that are commercially available, such as sclerolite or sclareol. And after that, we'll build the C and D ring of the target molecule through, I guess, contemporary methods in organic chemistry. We performed a little bit of enzyme engineering to optimize the hydroxylation of sclareol and sclerolite, and we were able to get very good conversion and exclusive C3 selectivity in all cases. And this is complementary to chemical approaches that always goes to C2, which is the init oxidation sites for sclerolite. Now, taking advantage of this platform, we can then come up with modular enantioselective synthesis of 8 different oxidized terpenoids. And we can do this in less than 12 steps, highlighting the versatility and also the enabling power of this strategy. So after this, we decided to set our sights on different types of terpenoids. At Scripps, this is commonly known as the wall of benzene slide that's designed to overwhelm you with the sheer number of structures. In this case, the take home is that all these 3 families, the N-corins, the N-athasanes, and N-trachelobanes, are very large and diverse. They contain many different members. And you might also note that the N-corin, the N-athasane, and the N-tracheolobane are rather similar in structure. They only differ in the identity of their CD ring, where N-corin has this 3-to-1 bicycle, N-athasane has this 2-to-2, and N-tracheolobane has this 3-to-1-0 tricycle. Now, they actually share a common precursor biosynthetically. They all arise from N-copolyl pyrophosphate. And it's commonly thought that there will be an initial cyclization reaction that cyclizes N-copolyl pyrophosphate to N-thimeranyl cation. This can then undergo different Wagner-Mirwin shifts to give rise to either the N-corin, N-athasane, or N-tracheolobane skeleton. And after that, tailoring enzymes would then take over, and this can happen in the form of direct oxidation or oxidative rearrangement to give rise to many different members of the respective families. Now, why do we care about these natural products? It was reported in 2016 that one member of the N-corin family, Quingensin A, is a unique inhibitor of heat shock protein 90, and the mechanism is as follows. So it's shown that Quingensin A can inhibit a novel site of HSP90, and this can then destabilize this complex that it forms with its client kinase and a co-chaperone CDC37. And the net result is that the client kinase would then be released, and this can be susceptible for degradation. And more recently, another member of the family, Oridonin, was shown to be an inhibitor of NLRP3 inflammasome. You might have come across this article at CNE News about the potentially useful application of inhibiting this inflammasome for tackling a lot of common diseases. The idea is that this NLRP3 inflammasome, when activated, can form a hexamer that in turn can activate caspase-1 and also cytokines such as interleukin-1b, and this would then trigger a feedback loop essentially of inflammation. And one common mechanism between the 2 natural products is that they both contain this enone as a Michael acceptor that can engage reactive cysteine in the proteomes. So the idea is that if you can gain access to many different members of the family, you can potentially develop novel covalent inhibitors or probes for biological discovery. And for this reason, as well as the interesting architecture of the natural product, there have been a lot of synthetic approaches towards these molecules, but unfortunately, I don't quite have time today to show all these approaches. I'm just going to focus on prior semi-synthetic approach. The minimally oxidized version of N-chorin is actually readily available. This comes in the form of stevioside, which is the main ingredient of your artificial sweetener stevia. And we're not the first to come up with this idea to use stevioside in semi-synthesis. The Baron Lab in 2014 developed this nice access to isoatazine in 13 steps from steviol. However, if you look at the synthesis, you will realize that the types of chemistry that you can do on steviol is quite limited to directed oxidation, starting from this C20 acid or alcohol, and also alert oxidation by taking advantage of this olefin here. And steviol does not contain any useful functional handle for functionalization at other positions of the molecule. And furthermore, it also has this olefin that can potentially be incompatible with a lot of oxidation methods. And to date, I think there is only one example of performing remote oxidation on similar scaffold, and this can be found on electrochemical oxidation of isosteviol methyl ethyl ester here. Now we realize that if we can come up with useful methods to functionalize either the B or C or A ring of steviol, we will be able to gain access to a wide range of members of N-chorins. And that will be the topic of this talk that I'm going to show you today, that we can start from steviol and access a total of 9 different natural products. And this can be done by a hybrid oxidative approach that combines chemical and enzymatic oxidations. And you will see later on that the oxidation step that we perform can also enable further skeletal rearrangement to gain access to different types of architecture. And in total, we made 9 different natural products. Each of them can be made in 10 steps or less, highlighting the enabling nature of this strategy. In terms of the oxidation itself, this is the, I think, the blueprint of what we want to achieve. If we can introduce hydroxyl group on the A, B, or C ring of N-carinoic acid or steviol through enzymatic means, we can then use this as functional handles for functional group interconversion to introduce more hydroxyl group. And we can also combine this with directed oxidation approach to quickly gain access to highly oxidized members of the family. Okay, this is a biocatalytic talk. So let's do a quick survey of what's known out there. The C7 hydroxylase is actually known from gibberellin biosynthesis. However, this enzyme not only catalyzes hydroxylation at C7, it also catalyzes further oxidation to give rise to this rearranged product that contains the gibberellin structure. So this won't be very useful for site-selective hydroxylation at C7. There have been quite a bit of investigations into using whole cell fungi for this type of transformation, but this is typically non-selective. You'll get oxidation at many different positions. And likely this is because fungal species would contain multiple P450s, and each of these P450s can have different selectivity profile, giving rise to a mixture of products. Luckily for us, our next door neighbor here at Scripps, the Shen Lab, has been investigating the biosynthesis of platensimycin. And they showed that the biosynthesis starts with this N-chorinal structure that can undergo initially oxidation at C11 with PTMO5. This is a P450. This is then followed by a cyclization step to form this gauged ether motif. In the next step, an alpha Kg enzyme, these two are quite similar in sequence, PTMO3 and PTMO6, and they both can catalyze hydroxylation at C7. And this C7 hydroxy group can eventually become the enone moiety of totensimycin after oxidative fragmentation of the A ring. And when we look at this pathway, we realize that PTM05, PTM03, and PTM06 can potentially be useful for our intended use. So we decided to investigate their synthetic utility. We start with PTM06 because it's the simplest to investigate. It does not require any dedicated reductase. We just simply test this in vitro hydroxylation with either steviol or N-corenoic acid. And we observe pretty good turnover numbers. And based on our previous experience, this is usually good enough for synthetic applications. For PTM05, I mentioned that this is a P450, and P450 requires a reductase coupling partner for electron transfer. And there is no reductase domain that can be found in the original biosynthetic gene cluster. So we have to play around doing artificial fusion with different reductases to determine what's the optimal chimera. And eventually we found that we can fuse PTM05 with RHF red. This is a reductase from a P450 from Rhodococcus. This gives the best conversion among the 3 reductases that we tested. I should also mention that we're not the first to use this artificial fusion with RHF red. For P450 application, David Sherman was the first one to demonstrate the utility of this fusion. And after that, we then perform a little bit of vector optimization as well as equalized strain optimization to bump up the yield to 88% on prep scale transformation. So now we've solved the oxidation of the B and C ring of N-corenoic acid or steviol. Time to turn our attention to oxidation of A ring. Since we have had some luck with using P450 BM3 variants for oxidation of terpenes, we decided to test BM3 variants in our collection for this purpose. And we were delighted to see that we can get some oxidation at C2, but for some of the mutants we tested, we also observed epoxidation at this exo-olefin. But fortunately, a little bit of alene scanning around the active site can tailor this site selectivity until we arrive at this variant called M177A that is selective for C2 hydroxylation without touching the epoxide at all. So now we have all the 3 enzymes. It's time to start doing the intended synthesis. We'll start with the synthesis of 3 natural products that contain mainly oxidation on the B ring. You can see that they have different oxidation patterns. So we want to show that by using one enzyme, we can access all of this different hydroxylation pattern. We'll start with mitricorinon, starting from N-corenoic acid that we can access in 2 steps from steviol. We perform oxidation with PTM06 to hydroxylate at C7. This can then be treated with DMP to the corresponding ketone. And then we found that we can treat this with pyridinium perbromide to affect intramolecular lactonization to complete our synthesis of mitricorinon in 5 steps with very high overall yield. Turning our attention to fuginoic acid, if we start from this ketone, essentially we have to oxidize this all the way to the dicarboxylic acid. And this can be achieved by first performing alpha oxidation of the ketone. While we were screening oxidants, we serendipitously found that PTM06 can also catalyze alpha hydroxylation when C7 is in the ketone form. And we found that this is actually a lot higher yielding than reactions using small molecule reagents such as Davis oxycyridine. So from here on, we can just perform oxidative cleavage with sodium pyridate and then treat this with DMP to complete our synthesis of fuginoic acid. And then finally, for farbosite A-glycone, we decided that we would dehydrate the olefin. We convert the acid to the metal ester that's required in the target molecule. And then we dehydrate the olefin, the hydroxyl group to the corresponding olefin, and then perform double the hydroxylation with osmium tetroxide to complete our farbosite A-glycone in 6 steps, again with high overall yield. Next, we decide to aim for targets that contain oxidations on multiple rings. We'll start with this target prostornin C and B. And if you look at the structure, what we need to do is to achieve hydroxylation at C11. C7 and C15, and then reduce that C19 from acid to alcohol or acetate. Now, at this point, we were faced with this dilemma, whether we should oxidize C11 first or C7 first. We can follow closely the native pathway of platensomycin biosynthesis by first performing C11 oxidation, followed by C7. However, if we do that, we would be facing potential issues when we want to convert the C7 alcohol from axial to equatorial. We can also think of doing the C7 hydroxylation first, and then convert it to the equatorial alcohol, and then try to do oxidation. But then now we'll be faced with potential selectivity issues when acetylating the alcohol to the this 11 acetate. So eventually we arrive at this decision where we would mask the C7 alcohol as the corresponding ketone in the synthesis. And this can be done by first performing hydroxylation with PTM06, followed by DMP oxidation. We give rise to this ketone. We can submit this for hydroxylation with PTM05 to introduce that C11 hydroxyl group. This can then be acylated with acetic anhydride. And at this point, we need to reduce this acid selectively without touching the acetate. And this can be done by first converting the acid to the acyl-imidazole and then add sodium borohydride in one pot that reduce this acid all the way to the alcohol without touching the acetate, and then also reduce the ketone to the corresponding alcohol. From here on, little oxidation complete the synthesis of prostonion C, and then further acylation complete the synthesis of prostonion B. And overall, this can be done in 7 steps. Next, we set our sights on fissurizin B. So fissurizin B contains this caged ether that's very reminiscent of platensomycin. And to do so, we first perform hydroxylation with PTM05 and then treat the product with TFA to affect the intermolecular cyclization. We need to remove this tertiary alcohol in a deoxygenation chemistry. In prior work, we've been using bromination-debromination sequence, but we found that this structure over here is actually inert for such sequence, so we had to come up with a workaround. And this can be done by first converting the free acid to the metal ester, and then subjected the product for Barton deoxygenation. After that, we can reduce the ester to the primary alcohol with diol, and then submitted this molecule for alkoxy radical-based functionalization to produce this iodoaldehyde that can then be oxidized and cyclized further to fissurizin B. And this can be done overall in 9 steps. Okay, now we've synthesized quite a few different endcorins. Let's start setting our sights on more complex targets. And here we aim to synthesize the mitraphorons. And if you look at the structure of the starting material and the product, you might think that I'm quite nuts in trying to get this to work. And admittedly, this requires a few lucky bounces to go our way. Much like this now famous shot from my favorite NBA player, Kawhi Leonard. He's my favorite player because I really aspire to his demeanor and career trajectory. And you will see later on how all of this lucky bounces would play out. The overall idea is that we're going to start from structure from, my mouse is missing. Oops. So we're going to start with steviol and then try to convert it to that structure down below. That is an N-baryon structure, and this is known through a rearrangement of steviol to isosteviol. And starting from this N-baryon structure, if we can generate a carbocation at C12, we can potentially effect a 1-2 shift that would generate an N-atthysane product. And from this N-atthysane product, we can if we can come up with a way to do a cyclization between that C13 and C16, we will arrive at a minimally oxidized N-trikylobin product that we can then submit it to all of the oxidation processes that we've developed before to gain access to all these 3 targets. Okay, this is the first lucky bounce that went our way. when we were hydroxylating isostabiol with PTM05, we found that it actually oxidized at C12 instead of C11. And with this, now we have the necessary functional handle to effect the rearrangement. We can do this by treatment with sulfuric acid. This affects a 1,2 shift to generate an N-atrazine product. Now to cyclize the C13 and C16, we first reduced that ketone to the corresponding alcohol, and we found that we can treat this with Lewis acid in the presence of silane to generate the desired N-tracylobane product. We think that this happens through formation of a non-classical carbocation structure that is then quenched selectively by the hydride at that carbon to the N-tracylobane product. Now that we have that, we can now begin the oxidation fun. We'll start with mitraphoron C. We first performed C2 oxidation with the BMP variant that we've developed before. We found that at high enough enzyme concentration, we can actually affect further oxidation to the corresponding ketone. We then submitted the product for hydroxylation with PTM06 and then do further oxidation with PDC to generate that diketone in the middle. Now we again take advantage of the promiscuous reactivity of PTM06 to produce that alpha hydroxy ketone that we can then methylate with diisomethane and oxidize further with DMP to generate the disphenol motif of mitraphoron C. For mitraphoron A, we can also perform similar sequence to get mitraphoron B in just 4 steps. I should also mention that we also attempted a different sequence where we first perform hydroxylation at C7 and then try to dehydrate to the olefin, followed by direct oxidation to the diketone, but this was found to be rather low yielding and accompanied by formation of that enone on the right-hand side. So we decided to go with the longer route because it gives higher yield. So now we have mitraphoron B. We need to convert it to mitraphoron A. In the previous synthesis of mitraphoron A, Tommy McGower has shown that you can do that final oxidation using either electrochemistry or Christian White's iron PDB catalyst. But we found by accident that if we just leave mitraphoron B standing in the presence of air, it can actually undergo slow auto-oxidation to mitraphoron A. And after 14 days, we can get 65% isolated yield of mitraphoron A. And this completes our synthesis of mitraphoron A in just 9 steps with 13% overall yield. Okay, so to conclude, I've shown today that we can start from stabiol and access 9 different natural products, highly oxidized natural products in a very efficient fashion by using a hybrid oxidative approach that combines chemical and enzymatic oxidations. And I think the enabling nature of the strategy is highlighted by the fact that we can make all of these natural products in 10 steps or less. And we think that now we have a very good platform to access a wide-ranging members of all of these natural product families. And we're excited in exploring that in the future. Okay, so it remains for me to thank people that are involved in this project. The main driver behind this project is Xiao Zhang. He's a very highly talented postdoc in my lab coming from Nankai University, and he was joined in this project by Emma King-Smith and also Li Cheng Yang. I also would like to thank my collaborators in the Shan lab, especially Jeff and Liao Bin, who initiated the collaboration. I would like to thank all of the funding sources, especially NIH, and I'd like to thank you all for your attention, and I'll be happy to answer any questions.
And a great talk, very impressive indeed. And I'm your round of applause.
Thank you.
So yeah, excellent. So we got a few questions.
Okay,
So first of all, we go on. You mentioned David Sherman, so maybe we should start with him. So
okay,
he says, very nice talk, Hans. Thank you. Are you pursuing structural studies on your P450 and alpha KG enzymes to facilitate future protein engineering studies to modify selectivity?
Yeah, that's a good question. The Shen lab actually is actively exploring that direction. They actually already have published crystal structure of PTM06, and I believe that they have ongoing work in trying to crystallize PTM05. And I think I agree with David that this can enable future engineering studies on these enzymes.
Okay, great. Another question from Nick Chiappini from Princeton University. Great talk. For potential of whole cell fungal biocatalysis, can you overexpress the desired P450 to a degree such that it overwhelms the other endogenous P450s and fungal oxygenases?
Yes, I believe you can, but I think the problem with this whole cell fungal biotransformation that I show is that they don't know which are the P450s that are responsible for the transformation. But in theory, if you know what P450 is involved in the reaction, you can overexpress that.
Okay. And then one other question from Nigyan Don. Thank you for the talk. I'm wondering if the chemoenzymatic reactions is done under aqueous conditions. The substrates of those reactions don't seem to really like water. So I'm wondering if solubility of substrate has ever been a problem to your reaction.
Yes, good catch. We did everything in aqueous buffer. We use DMSO as our co-solvent. We use 5% DMSO in all of our reactions. And we've gone up to 10 millimolar substrate loading, depending on the reactivity of the enzyme. In terms of solubility, we do see when we add everything to the mixture, we'll end with a suspension. But I think that all that matters is to have a little bit that dissolves and form the product, and your Le Chatelier equilibrium would then eventually drive the equilibrium towards the product formation.
Okay. Then we got a question from Therindu Ranathunge. How do you monitor these enzymatic reactions?
We typically just let them run overnight, to be honest. I think that's one of the beauty of enzymatic reactions. They are very selective and mild that you don't have to worry about side reactions. You can also, if you want, you can also TLC the reaction. We've done just taking the flask that contained the reaction suspension, drop in a TLC spotter to the reaction and spot it on TLC plate. That works too.
Okay. So a question from me. I'm tremendously impressed with what you've achieved here and the way you're controlling the selectivity so nicely by choosing the right enzyme. But how easy is it to, you know, to get that enzyme to work? In other words, to get the enzyme you need to be able to scale it up in an appropriate way. You know, obviously there's lots in the audience that are like me who come from more of a traditional synthetic background. Yeah. So maybe you can give us a little bit of an introduction of how easy it was for you to do this.
To get the enzyme you need, there are many companies out there that can do DNA synthesis for you. So if you have the protein sequence, you can simply order the requisite expression vector to express the enzymes. And from there, you can just introduce those vectors into E. coli or other strains or other organisms of your choice, and then do that to express your proteins. Sometimes you have to play some tricks to optimize the enzyme expression. Some enzymes just don't express very well, and I think it's all empirical at this point. You just have to try and see whether they express well or not.
I see, great. Well, Hans, thank you. A very impressive talk.
Thank you, Genju.
It's very impressive. Congratulations. Okay, so our third speaker is Michael Hilinsky. So Michael did his BS at Tufts University, and then did his PhD at Stanford University with Paul Wenter. Then he spent a couple of years in industry, and then had a postdoc at the University of Virginia, and must have impressed people during his postdoc, because he moved directly then into being an assistant professor at the University of Virginia. And he's been interested in organocatalytic approaches for doing CH oxidation and CH amination, and has achieved a really interesting range of transformation. So I'm delighted that we have him speaking as our final speaker today. on organocatalytic approaches to atom transfer CH functionalization. Welcome, Michael.
Thank you, Hugh, for the introduction and for the invitation to speak among this great group of speakers today, and for building the infrastructure so that we can actually give talks and listen to talks during this time. So it's very a lot of foresight there on your part and on the part of the whole center. So yeah, what I would like to tell you about today is some of the things we've done in recent years, looking at organocatalytic approaches to atom transfer functionalization. And, you know, in particular, we're interested in developing new ways of accessing catalyst control of site-selective oxidation of alkane type CH bonds, sp3 hybridized carbon-hydrogen bonds. And if you look at the literature historically and in recent times on this kind of transformation, in terms of small molecule catalysis, not to take anything away from enzymatic catalysis, has really been focused on a transition metal catalyzed approach. And there have been many groups working in this area, many of which I've tried to capture here on this slide. And of course, as we all know, these typically go through mechanisms where you're accessing from the catalyst either a metal oxo or a metal nitrenoid type of intermediate, which will do CH insertion type of reactivity. And so we, when we were taking a look at the scope of work that had been done on these kinds of transformations, I think we identified that organocatalysis was a relatively underdeveloped approach and may lead to discoveries of new reactivity and new types of selectivity in this area. And I'll talk a little bit more about the chemistry involving this, but these would typically involve oxidants, at least through the approach that have been used before and that we've continued exploring, of these three-membered rings that contain weak heteroatom bonds. And there were examples of organocatalytic atom transfer hydroxylation in the literature, but not of amination. And so we really identified this as an unexplored area in the field. And you know, why are we interested in trying to develop a new platform for this type of small molecule catalysis? I think, you know, in my mind, the problem is captured by examples on this slide. For example, one can take a complex molecule like artemisinin, and one can take an iron catalyst and get a site-selective hydroxylation of a tertiary CH bond, as shown in this slide. And similarly, one can take a transition metal catalyst in this case, a rhodium catalyst, and do a secondary CH amination, forming that protected amine in a stereoselective manner. And these reactions are quite useful for late-stage diversification of bioactive compounds. The issues being, though, that most of these are substrate-controlled reactions at this point, and we might be able to use catalyst control to access all of the other carbon-hydrogen bonds on this molecule that are not currently able to be oxidized in this manner. And so really there are several challenges that might be addressed by new mechanisms and new approaches for these types of atom transfer CH oxidation reactions. And so here's some of the prior work before I get started into what we've contributed. You know, and this was entirely work done by the Dubois group in terms of these organocatalytic CH hydroxylation reactions, based on the observation of some earlier groups that one could take sufficiently electron poor oxyuridines of the type shown on the top here, and affect these site-selective CH hydroxylations that occur stereospecifically with retention of configuration, and generally go for the most electron-rich carbon-hydrogen bond on a molecule. I think, you know, what the Dubois group recognized was one could potentially access a similarly electron poor oxaziridine via a catalyst, in this case benzoxathiazine catalysts, and they were able to affect these transformations using an organocatalytic approach, and they developed this over several different papers. And so I think this really gave us some tailwinds that maybe this could be accomplished in terms of other types of transformations and using other types of catalytic platforms. And so I think where we come in is just thinking about these types of oxidants in a little bit broader sense in terms of atom analogy. One could imagine, as was shown in the previous slide, doing imine catalyzed reactions that go through oxaziridine intermediates. But in a related sense, one could think about doing ketone catalyzed hydroxylations from via dioxiranes, or using the cationic versions of the imine type of catalyst, an iminium catalyst in this particular case. And also by flipping these oxidants around, and imagine now transferring nitrogen via ketone or iminium catalyzed reactions. And I won't have time to talk about all the work we've done in this area today. You can read about it in this Syntax article we published a few years ago. Some of our early work, oh, excuse me. Some of this early work, our early work has been focused on developing the first ketone catalyzed CH hydroxylation reactions. And again, you can read more about that in that Syntax article. But what I wanted to talk about today was really our work on iminium catalyzed reactions of this type. And sort of, you know, one of our long-term goals in terms of this type of chemistry has been to take organocatalysis and enhance the flexibility in terms of the different types of atom transfer reactions one could accomplish, in the same way that one can take a rhodium catalyst and accomplish both CH hydroxylation and CH amination reactions with some tweaks to the catalyst structure. So this was our goal, our long-term goal for this type of chemistry. And so you could imagine in the presence of a suitable oxidant from an iminium salt making an oxaziridinium, or in the presence of a nitrene precursor making a diaziridinium. And the question here was, you know, could we access these types of oxidants in a catalytic fashion to affect these site-selective CH oxidation reactions? The sticking point here being that neither of these species, unlike some of the dioxiranes or the oxaziridines, had been shown to do this chemistry even in a stoichiometric sense. So there was a bit of an unknown there. You know, one attractive part of working on this type of project was, you know, at least for oxygen transfer, for epoxidation reactions, there's a sort of a wealth of literature on an enantioselective chiral catalyst that can enable enantioselective epoxidation reactions and other types of reactions such as sulfide oxidation and so on. And we thought that might, if we could solve this catalysis problem, we could start to adopt some strategies developed by others to do asymmetric enantioselective catalysis. So we decided to start with the oxygen transfer reactions, since this epoxidation reactions had been reported before in the literature. And some of our first forays into this were taking this isoquinolinium catalyst that had been previously shown to affect epoxidation reactions. And here what we're looking at is a typical screening substrate where we have say an electron-rich tertiary CH bond versus an electron-poor CH bond. So we're looking at reactivity and selectivity in this manifold. With this starting scaffold, we saw essentially no reactivity whatsoever. If we add a trifluoromethyl group to this scaffold, then we start to see the required reactivity. The other thing we were seeing for these reactions was that we were getting a lot of catalyst oxidation, as you might expect, and so we were able to suppress that by adding a gem dimethyl group at the benzylic position of the molecule, and that allows us to get a catalyst that is stable enough to then give us reasonable conversions and yields for these hydroxylation reactions. So I'm not going to belabor the substrate scope too much here, other than to say that the yields here are isolated yields. The yields shown in parentheses are based on recovered starting material yields. We'd never see full conversion for these catalysts for reasons that I'll explain in a moment. These reactions are stereospecific, and they follow all of the types of selectivity rules, going for the most electron-rich CH bond in the absence of steric effects that you would expect. One serendipitous discovery that we made in the course of this project was that we discovered that when we have unprotected primary alcohols or primary ethers, as in this particular case, we see no alcohol oxidation. We see exclusively aliphatic CH hydroxylation in this particular case, which was quite surprising to us. This selectivity extends to selectivity for alkane hydroxylation over secondary alcohol oxidation as well. We see no ketone product in this particular case, even though the yield is low, and that's in part because we had to add a co-solvent to get this dihydrocholesterol to dissolve in these solvent system. For a simpler secondary alcohol, we get maybe 3-to-1, 4-to-1 selectivity when steric effects play no role. This extends to selectivity for hydroxylation products over ketone products of over-oxidation. In other words, when one is oxidizing a secondary CH bond, cyclohexane gives about a 6-to-1 ratio of cyclohexanol to cyclohexanone. And so we investigated this initially and found that solvent effects were playing a key role for this. And so the current understanding of this selectivity, as also furthered in some work a year after us looking at transition metal catalysts, is that it's the strong hydrogen bond donating ability of this hexafluoroisopropanol solvent that deactivates HA versus HB and HC and directs selectivity to this site rather than alcohol oxidation. And so here's a little information about what we view as being the catalytic cycle for these reactions. So hydrogen peroxide is our only oxidant and we only reagent besides solvent and catalyst and substrate that's required. And so we envision that this forms a peroxyaminal. We can observe that by NMR. That liberates a equivalent of Bronsted acid, which then allows us to close down to an oxyzerodinium salt, which we believe is the key species that is affecting the CH hydroxylation reaction, and we have evidence of that being the key oxidant. One thing that we do observe is that there's a liability to this catalyst, which is that once you form the oxyzerodinium, you can deprotonate at the adjacent position here to form this alternative aminium, which is not a great catalyst for the reaction. And so that's why that's what kills our turnover and our conversion for these reactions. And so that's a problem that we're currently working on to solve. But what I wanted to tell you more about in detail is how this got us into looking at the first examples of organocatalytic atom transfer CH amination reactions. And this came from the simple idea that if one could do this with oxygen, one could similarly take a nitrine precursor and in a similar way access a diazerodinium as a putative oxidant. And the question here was really, would this diazerodinium be capable of doing a CH insertion and forming a CH amination product and with catalytic turnover. And really this approach for organocatalytic nitrine transfer, even in other contexts such as azerodination, had not been explored. And so we decided to go down this path. And the problem being, though, that there was very little information about these diazerodiniums in the literature. Here are some computational studies that we found from the Hauk group, showing reasonable barriers for aziridination of olefins from a diaziridinium species. So that was really the only computational paper we could find, and the only experimental study of any value was really from the Lambert group, which had they had synthesized and unambiguously characterized by crystallography these NH diaziridiniums. And of course, they were interested in testing these in aziridination reactions, but they never observed the aziridine. Instead, they observed this ring expansion reaction. And so we were optimistic that we could overcome this by the use of a protecting group on the nitrogen, which would of course reduce the electron density here and hopefully bias the reaction toward nitrine transfer over the ring expansion reaction. And in fact, that's what we found pretty readily once we took the same iminium catalyst that we had used, that we had developed for the CH hydroxylation reactions, using this pentosyl iminoiodinane as the nitrine precursor, we can get substrate limited CH amination. And so this works for benzylic positions. If you have a strong driving force for overoxidation, you get the imine products rather than amine products. It works for some of the more reactive alkane substrates as well, for example, adamantane. And it does exhibit site selectivity on more complex substrates, which I think is really indicative of potential future promise of this type of organocatalytic reaction. There are some limitations to this. I will say that it's hard to get away from benzylic amination and do more alkane types of substrates, but we are hopeful that this can eventually be addressed by catalyst design. And so I'll tell you a little bit about our thoughts on the mechanism of this reaction. Since the approach was so new, we really envision this as forming some sort of organic nitrinoid that would, such as the diaziridinium, that would do a CH insertion reaction. So we've ruled out some other possibilities, such as oxidation of the substrate, say in this case to an oxocarbenium ion, and then nucleophilic addition of a free sulfonamide that was liberated from the nitrine precursor. And we've done a competition experiment which rules that out in the case of isochroman. And other information about the mechanism that we've gathered are from, for example, these KIE experiments where we see a small but primary kinetic isotope effect, indicating that CH bond cleavage is occurring in the rate determining step or in a product determining step following the rate determining step. So if we're making an organic nitrinoid, we should expect this to perform other types of reactions, including aziridination. And so we've recently published last year some of our initial studies on this reaction. So right now we're limited to being able to aziridinate styrenes, but we do see some reasonable functional group tolerance, such as the boronate and acid sensitive groups such as the TBS group here. One can imagine that we specifically chose this vinyl tetralin substrate as a probe for selectivity between CH amination and aziridination reaction. And as you would expect, based on one's predictions for a nitrinoid type of species, certainly the pi bond is more reactive in this particular case. So that's all consistent with our working hypothesis. Some interesting mechanistic information that came from this aziridination, and I'm getting away from CH bonds for a moment here, but I think this is relevant to the mechanism of the CH amination, which I will describe momentarily, is that this reaction is completely stereospecific, but for the trans product in each particular case, we get exclusive generation and a complete inversion of geometry from the cis beta methyl styrene in this particular case, which is unusual and of course suggesting that we're going through some sort of long-lived intermediate. And this particular product doesn't arise from the cis-aziridine at all. That's completely stable to the reaction conditions. Some other things that we observed is that under modified conditions, we can get stilbene aziridination to proceed. However, if we use the conditions at higher temperature and with a little bit different solvent that we optimize for aziridination reactions, we see evidence of this 1,2-phenyl shift generating this enamine product. And one thing that my student, Freddie, kept coming to me about was that every time she was doing these reactions, she would isolate the aziridine product, but then she would also isolate a small amount of this imine product out of the reaction. And we thought about this for a little while until we started thinking about it in the right way, which is as an oxidative cleavage of this olefin. And so in an oxidative cleavage, you would expect to also see the formamine as a secondary product of this reaction, but we weren't able to fish that out. And so going to a higher molecular weight, sort of R2 group on this side rather than hydrogen, we do see indeed, in addition to sort of a low yield of the aziridine product, evidence of oxidative cleavage. And so that led us to propose this particular mechanism for the aziridination reaction, where we're going through, again, the postulated diaziridinium species, a 2-electron attack of the olefin, which would explain our limitation to styrenes at this point, would generate a carbocationic intermediate, long-lived enough for bond rotation. Ring closure would give us the aziridine. When your R2 group has a sufficient enough migratory aptitude, you would get shifts of that group. And if you bring in another equivalent of the amine, the nitrene precursor in this case, you could imagine this adding in and through the arrow pushing shown in blue, leading to the observed oxidative cleavage reaction. Now, in the last little bit here, I want to talk about some more recent studies we've done in collaboration with the Gutierrez group at the University of Maryland, trying to get at a better understanding of the mechanism of the CH amination reaction. So again, what we're proposing is that we would make a diaziridinium, and that diaziridinium is the active species for site-selective amination. And here's the data that shows that this is indeed a catalyzed reaction that is specific to the aminium. So the uncatalyzed reaction doesn't proceed for amination of tetralin, nor if you just add a normal phase transfer catalyst such as the tetra and butyl ammonium tetrafluoroborate, you also see no reaction. So this is not acting as a phase transfer catalyst in these reactions. And so in collaboration with the Gutierrez lab, we basically considered 3 different potential active species, the diaziridinium, the diradical, and this nitrenium species down at the bottom. And we imagine that either of these could ultimately undergo one of 3 paths for CH amination, either a concerted insertion, a hat radical rebound type of mechanism, or a hydride transfer nucleophilic attack mechanism to generate the observed amination product. And so I'm going to skip a lot of the calculations, and I'll read to you here what is not shown. So one of the things we do find is that the diaziridinium is, as one might expect, a thermodynamically favored intermediate along the reaction pathway. And we also calculate that the rate determining step is formation of one of these 2 species. So this D, the triplet D or the D, and I'll talk more about those in a minute. So that's consistent with the KIE experiments that I explained earlier. And so interestingly, the D and triplet D, so the singlet and triplet forms of this diradical intermediate are isoenergetic. And so we considered CH amination from either of these intermediates. For other intermediates along the pathway, such as the diaziridinium, the calculated barriers were just way too high. So interestingly, from the singlet form, we observe that the calculated mechanism is a hydride transfer to get to this carbocation intermediate, followed by a nucleophilic rebound. From the triplet form, another mechanism dominates, which is HAT. But interestingly, we don't calculate that the radical pair, or that the radical pair then undergoes a radical rebound, but rather that electron transfer ensues, and that you get carbocation intermediate, again getting you to this nucleophilic rebound type of mechanism. And so, you know, that's all captured here in this catalytic cycle. So again, here the triplet biradical would go through this HAT followed by intersystem crossing to get you back on the same catalytic cycle as the singlet biradical with hydride transfer, or rather, in some cases, this carbocationic intermediate dominating. And that's consistent with Hammett studies we've done that show a fairly strong dependence on this R group in terms of the observed rates in a competition experiment, with strongly electron donating groups like methoxy giving high yields of products, and acetyl, for example, giving us essentially no reaction under these conditions. And again, I think this hydride transfer, I think showing the electrophilic nature of these intermediates, leading directly to perhaps a carbocation in the case of these examples I've shown, is consistent again with that aziridination reaction proposed mechanism, which also goes through a 2-electron process. So, you know, I hope I've been able to sufficiently describe, you know, some of our interesting findings recently on these aminium catalysis of reactions. And I think, you know, we've continue our efforts to sort of fill in these gaps in terms of organocatalysis of these reactions. And, you know, ultimately, in what we're currently pursuing is using catalyst control, now that we've solved the fundamental catalysis issues on these systems, to really exert control over things such as site and stereoselectivity, functional group tolerance, etc. And so here's my acknowledgements page. So as you can see, this is an old picture. We haven't, in part because of coronavirus, been able to take a new group picture yet, but the first years have photoshopped themselves in. This work was done primarily by former group members Logan, Balaram, Will, Daoyang, and continues to be, and Rob helped out with the recent collaborative paper, and continues to be pursued by others in my group. I'd like to thank our collaborator and all these institutes for funding, especially for this project, the NIH and the ACS PRF.
Okay, Mike, thanks very much for a great talk. And here's my individualistic clap.
Thank you.
Okay, we got a couple of questions. One is from Nick Chiappini from Princeton University, and he's got previous experience, I think, working with Justin Dubois on metal nitrine chemistry. So first of all, awesome talk, Mike. On the aminium catalyst for your N and O transfer chemistry, have you explored longer or bulkier perfluoroalkyl substituents?
Yes, indeed we have. And so far, at least for the types of reactions shown, we found that trifluoromethyl is still the best.
Okay, great. And then we got a second question. I think this is from Shannon Stahl's group. Do you have any clues if the catalyst decomposition is first order or second order in catalyst for the hydroxylation reaction?
That is a very good question, and no, we haven't looked at that. We haven't looked at that yet. No, we don't know.
Okay, great. Well, those are the questions at this stage. At this stage, I'd like to thank our three speakers for been willing to speak at these challenging times. A lot of people are working a lot of details with their teaching at the moment and organizing their labs in different ways as well. We have, oh, actually, there are tons of questions for you coming up. Wow, I only saw 2, but there's actually 12.
Okay, I can get on that typing away for at least the next little bit.
Yes. And thanks very much for the great participation too. We've had so many great questions during the event. We've had close to 600, if not even more, joining the meeting. And again, let's thank our 3 speakers for such an exciting selection of talks, really showing all the interesting different ways that CH functionalization is still developing. So again, thanks very much to all of you. And hope we'll see you again at our next meeting in, I think, was it May 30th? Well, we'll send you notices in due time. Okay, bye now, everyone.
All right, thanks, Hugh.
Bye.
Mike, I'll keep the meeting open so that you can continue to answer those if you would like.
Okay, thanks. Yeah, it's like 10 minutes or something.